Alright, so hopefully you're in the right spot. This is an anatomy lecture for my students at Long Beach City College. This is for Chapter 10, which is an introduction to the muscular system. You can find this PowerPoint file in Canvas, and as well you can find the notes to fill in. I'll probably go through the PowerPoint faster than I normally would because I will assume you guys are pausing it to write in the video. Let's first review Chapter 9, which was the last time we met. The term diarthrosis refers to a joint that is freely mobile. So diarthrosis is freely mobile, letter B. The sagittal suture is an example of a fibrous joint, letter B as in boy. An articular capsule is present in synovial joints, letter C. The interosseous membrane between the radius and ulna is an example of a syndesmosis, letter E. What type of cartilage is located between the bones and the pubic symphysis? This is fibrocartilage. The pubic symphysis keeps your pubic bones together. Which is not true about synovial joints. They do have articulating bone surfaces covered with cartilage. They're enclosed by an articular capsule. There's synovial fluid secreted by the synovial membrane, and ligaments strengthen and reinforce the joints, but you do not have blood circulating within your joint cavity. So letter E is the answer to this one. You will have blood vessels associated with a joint, but blood circulating within the joint cavity would probably be a cause for alarm. There should only be synovial fluid there. Which are not considered to be features of a synovial joint. They will have nerves, they will have blood vessels, just not secreting blood into the cavity. There will be articular cartilage, and there will be synovial fluid. We don't usually clump muscles into synovial joints, so muscles aren't a part of a synovial joint. The joint formed by the axis and atlas that permits the no shaking of the head, that's an example of a pivot joint, letter D. And rotation of the forearm so as to direct your palm anteriorly as an anatomic position. You can picture me up in the front of class, standing there. My palms are facing forward. I can hold soup in my hands, so we call this supination. So rotating your forearm to direct your palm anteriorly is supination, letter D. Movement of a body part toward the main axis of the body is called adduction, letter E. So adduction, you're adding a body part back to the midline. This is in contrasting to abduction, where you're abducting a body part from the midline. So again, those are review questions. Your test will include more questions like that, but if you know the answers to those and understand what the answers mean, that'll help you for your next lecture exam. So this is Chapter 10, Intro to the Muscular System. Here's our outline. Again, I go through this a little bit faster, faster than what I would normally do in class, because I will assume you guys can just pause the YouTube video if you need to. Your first length is that there are over 700 skeletal muscles, and together they form the muscular system. We won't be learning all 700 in your lab exam. More generally, there are 3 types of muscle tissue: skeletal muscle, cardiac muscle, and smooth muscle. So those are your 3 types of muscle tissue that you can write in your notes: skeletal, cardiac, and smooth. And we'll focus a lot on skeletal today. The properties of skeletal muscle tissue, all of them, all the muscle tissue types are excitable. That means they can respond to a stimulus via your nervous system. They have conductivity, which means they're able to transmit electrical events along the cell membrane. Their ability to contract allows them to generate tension and shorten their cell length. That is important because their ability to contract and shorten is what will allow them to pull on bones to allow you to move parts of the body. Their elasticity is important because that is what allows them to return to resting length after shortening or lengthening. And extensibility refers to their ability to be stretched beyond their resting length if needed. Each skeletal muscle is considered its own separate organ, so that means it will have all of the 4 tissue types within it. So your biceps brachii, for example, located on the anterior part of your upper arm, contains all 4 tissue types. They are striated underneath a microscope. You can see those zebra stripes. They're usually attached to bones, and their muscles and muscle cells can vary in shape and size. Some of the functions of skeletal muscle tissue, these are the ones that really help with body movement, maintaining posture, protecting and supporting organs that lie beneath them, and then regulating elimination of materials. They will make up some of the sphincters at your orifices as substances leave the body. They're also important for generating heat, and we'll learn about that when we get to nervous system. Skeletal muscle organization. You should know how muscles are organized within themselves. A bundle of muscle fibers is called a fascicle. And when we say muscle fiber, that means muscle cell. So this is where each cell in a muscle is called a fiber, and a fascicle is a bundle of muscle fibers. Those muscle fibers contain myofibrils, which are complex cylindrical organelles, and each myofibril is made up of myofilaments. There's a great video that you can watch on your own, and I have the link included here, so I'll let you guys watch that link on your own. But basically, it takes you through the organization of a whole muscle. So you can see the muscle at the end of the tendon. The whole muscle is made of fascicles. Fascicles are made of myofibrils, and myofibrils are made of myofilaments. So here we have a great chart detailing the organization of muscles. If we start going from large to small, each muscle is made up of fascicles. Each fascicle is made up of muscle fibers. Each muscle fiber is made up of myofibrils, and each myofibril is made up of myofilaments. So you should know that order in going from large to small. Then we talk about the layers of connective tissue that surround each of these structural organizations. These layers are connective tissue, and they provide protection, sites for blood vessel and nerve distribution, and attachments to the skeleton. So these connective tissue components, the endomysium, think endo means inside. Endomysium surrounds and electrically insulates each muscle fiber. The perimysium surrounds each fascicle, and the epimysium surrounds the entire muscle. Then we have deep fascia and superficial fascia, and the fascia will help separate the muscle from the skin and also just serve as packing material. A tendon then will attach a muscle to bone, skin, or another muscle. This is formed by the merger of connective tissues at the end of the muscle. It's a rope-like structure. It's different from a ligament. A ligament will attach a bone to a bone and help kind of keep a joint strengthened, such as your ACL, PCL in the knee, but a tendon attaches a muscle to a bone. An aponeurosis is a thin, flattened connective tissue attachment that we'll talk about when we see our muscles. Most muscles extend over a joint and attach to bones on either side of the joint. We call one end of the muscle the origin and the other end of the muscle the insertion. One end usually causes one bone to move, while the other bone or other end of the muscle and bone remains more fixed. The less movable attachment is called the origin of the muscle, and the more movable attachment is called the insertion. And knowing the origin and insertion are important in muscles because knowing where they originate and what bone they attach to will then help you figure out their action of that muscle, whether it flexes, extends, rotates, abducts, or adducts. We also call them superior or inferior attachments or proximal and distal attachments, but In this class for me, we will go by origin and insertion. So the origin and insertion, you can see here on the biceps, it originates or attaches to parts of the scapula and the humerus, and then it inserts itself on the radius. So you know that when that muscle contracts, the insertion will pull up on the radius and will cause flexion of the forearm. So then if we go to the second page of your notes, the microscopic anatomy of skeletal muscle, the sarcolemma is similar to a typical cell, but it just has a different name. So the sarcolemma is the plasma membrane, the sarcoplasma is the cytoplasm, and the sarcoplasmic reticulum would be what the smooth ER would be. If we take a look at two other main structures that are unique only to muscle fibers which help with their functions, the T tubules are deep invaginations of the sarcolemma that extend into the sarcoplasm, and then the sarcoplasmic reticulum is this internal membrane complex that actually stores calcium. And calcium is incredibly important for initiating muscle contraction, which you'll learn all about in physiology, and we're only going to touch on here. So what specifically are these myofibrils? They are cylindrical structures within each muscle fiber cell that run the length of the cell. They have the ability to shorten, which will result in contraction of the muscle fiber, and they will contain myofilaments, which are strands of proteins that allow for contraction. There's two types of myofilaments, thick filaments and thin filaments. And you should know what another name for thick filaments are and thin filaments are. And we'll show you this right now. Thick filaments are also known as myosin molecules, and each myosin molecule has kind of two strands to them, a tail that's intertwined, and then with a head. They look almost like golf club heads. Those heads are important because they form a connection with the thin filaments during contraction. The heads can actually move up, attach to the thin filaments, and then pull on those thin filaments as the thin filaments slide past them. So thick filaments are myosin, and thin filaments are composed of actin. Actin is a thin filament. It's made up of kind of two strands of actin that are twisted around each other, and there's two regulatory proteins within that thin filament. Tropomyosin is a thread-like protein that covers up a part of actin that's in green in that picture, and troponin attaches to actin and tropomyosin and can bind calcium. Pretty much what calcium does is calcium will bind to the troponin and move off the myosin binding site. So that myosin can then bind to the actin. So in the myofibril, the actin and myosin are kind of layered on top of each other. So the myosin are the orange structures with the golf club-like heads. Those heads will reach up and attach to thin actin filaments on either side of them. The thin actin filaments are shown as kind of that double-stranded bead-like structure. When the myosin attaches to those thin filaments, it'll slide the thin filaments past each other, causing the muscle to shorten or contract. And this brings us to what a sarcomere is. So a sarcomere is one unit within a myofibril, and we call this the structural and functional unit within a myofibril. A sarcomere is described as spanning from one Z-disc to the next Z-disc, and that's all you need to know. That's listed for you. The Z-disc is the jagged edge line in which the thin filaments will attach themselves to, and the Z-discs will shorten during muscle contraction as the actin thin filaments slide past the myosin. This sarcomere is divided into different bands. You see an I band, an A band, an H band, an M line. These names are important for describing the sliding filament theory and what exactly happens to the actin and myosin as they slide past each other. But what you need to know is that each sarcomere, which is the structural and functional unit, spans from one Z disc to the next. This slide describes the different bands of that sarcomere. The I band contains your thin filaments, but not your thick. Your A band contains only thick filaments, and the M line goes right down the middle. Here's what a sarcomere looks like and how it's arranged. And the description of all those bands, but I just want you to know what this sarcomere, that the sarcomere spans from one Z disc to the next. This sliding filament theory we'll get into a little bit here. There's a great video on it to show you what it looks like. The sliding filament takes you through the physiology of muscle contraction. Basically, to do this, muscles develop tension and their sarcomeres shorten. And for sarcomeres to shorten, your thick myosin filaments attach to your thin actin filaments and pull them towards the center of the sarcomeres. And the sliding filament theory explains what muscle contraction is, which, trust me, we'll learn in detail in physiology. What I want you guys to know for anatomy are some of the names and just have a general idea of what happens. So during contraction, thin filaments will slide past thick filaments and the Z disc will move closer together, causing the sarcomere to shorten. The lengths of the filaments themselves never change. Only their relatives' positions will change. And this is showing the sliding filament theory, showing in a relaxed muscle how the sarcomere is at resting length from one Z disc to the next. But in a fully contract muscle, you can see how the sarcomere shortened in length when everything slid past each other. The next slide has a video describing what rigor mortis is. And rigor mortis has to do with the stiffening of your muscles at death. So again, if you find yourself with a dead body in the woods, you can figure out how long it's been dead by how tense or stiff its muscles are. At death, your muscles are naturally stay in their contracted state because your muscles need energy for the myosin and actin to disconnect from each other. Without that disconnection from energy, or ATP, that is needed, your muscles will stay stiff. So that's what rigor mortis is. You can tell how long a body's been dead based on how stiff their muscles are. Eventually the muscles will relax, or not relax, but they'll just degrade and the body will become completely relaxed. So that is the video for rigor mortis if you want to watch it. Neuromuscular junction then is a more physiology that you'll learn, but it's the junction where a motor neuron impulse stimulates an impulse in a muscle fiber. You remember that skeletal muscles are voluntary, so your somatic motor neurons control the actions of your muscle fibers. The contraction begins when a motor neuron impulse stimulates the muscle fiber, and the neuromuscular junction is the place where the motor neuron communicates with the muscle fiber. There's some anatomical terms that happen here. The synaptic knob is the expanded tip of a neuron axon. The synaptic vesicles are membrane sacs in the synaptic knob that are filled with acetylcholine, and acetylcholine will travel across a synaptic place to the motor end plate, which is the region of the sarcolemma with many folds. Acetylcholine receptors bind acetylcholine and then continue to stimulate and send on that stimulus towards their muscles. So this is a look at a neuromuscular junction. Again, it's the junction between a nerve and a muscle, so it allows each muscle to control each muscle fiber. This really is tied to what a motor unit is. A motor unit is a single motor neuron and the muscle fibers it controls. A motor unit contains only some of the muscle fibers in an entire muscle. When a motor unit is stimulated, all the muscle fibers within it will contract, and the size of the motor unit and degree of control provided are inversely related. What that means is that small motor units provide precise control, such as in your eye muscles, and lower limb muscles have large motor units or many fibers because they require more force recruit from more motor units. So this is a look at a motor unit and one neuron controlling one specific muscle fiber. Muscle tone then refers to the constant tension in a resting muscle. Motor units are usually stimulated randomly to avoid fatigue of a muscle, and there are two types of muscle contraction: isometric contraction, where the length is constant of the muscle fibers but the tension is changing. In isometric contraction, there's no movement because the length of the muscle is constant. In isotonic contraction, you do get movement of the muscle. The tension will be constant, but the length of that muscle itself is changing. You should know the difference between isometric and isotonic. And I'll show you a picture of this elderly woman with gray hair, although it could be me. But she's trying to lift an 80-pound resistance weight, and her muscle tension is less than the weight of the resistance, so there's no movement. So this is an isometric contraction. In the second example, in isotonic contraction, her muscle tension is greater than or equal to the resistance, so there will be muscle movement. That's an isotonic contraction. Another example of an isometric contraction would be as if you're pushing hard against a wall. There will be no movement there because your muscle's tension is less than the resistance of that wall. Then we'll talk about types of skeletal muscle fibers. They can be slow or fast muscle fiber types, and an individual muscle typically has some of each fiber type. If we look through the differences between slow and fast fiber type, this chart is really helpful. We'll talk about color first of all. Slow muscle fibers are usually darker reddish in color because they have lots of blood capillaries, and fast muscle fibers are usually white or lighter in color because they have less capillaries going through them. They're called slow because of how fast they use ATP, so slow fibers use ATP slowly, fast fibers use ATP more quickly. Slow fibers have lots of mitochondria, and they're able to generate ATP really quickly, and fast fibers don't have as much mitochondria, and they're not able to generate ATP as quickly. So because of the number of mitochondria that's in each muscle fiber type, slow muscle fibers are typically more for endurance, like marathon training. The muscles in your back, your muscles in your trunk, the muscles in your legs have a lot of slow fibers in them because they help with endurance, maintaining posture. They have to be contracted all the time, or if you're standing all the time, they have to be contracted. They're also darker red in color. Then your fast fiber types are more for short duration stints or intense movements like sprinting or lifting weights. So you'll find fast fibers in your upper limbs. Fast fibers are white or lighter colors, so this applies to the type of meat you like to eat in chicken or turkeys, whether you like to eat dark meat or white meat. The dark meat that you're eating is a slow fiber muscle meat, and it's darker in color because of the extensive blood capillaries. And where is dark meat found in a chicken? It's found in the legs and in the wings, which are really what the bird is using for endurance. And the fast fibers are the white meat in the breast. They're not used for endurance, they're used for short durations. So that's kind of the white and dark meat comparison in poultry or chicken or turkey if you eat meat. So this is a look. SO stands for slow oxidative, FG and FO stand for fast oxidative and fast glycolytic. This just shows that each muscle has kind of an assortment of all different fiber types depending on what it's useful for. And again, depending on where the muscle is, there will be more types of fibers of one type than another. And this just describes what we just talked about. A skeletal muscle usually contains all three fiber types. Slow fibers dominate postural muscles, such as those in the back and calf. And there are no slow muscle fibers in muscles that require swift but brief contractions, such as in the eye and the hand. So, to how skeletal muscle fibers are organized. They are organized into fascicles, and the fascicles can have different patterns of arrangement throughout the body. They can be arranged circularly, so they can go in a circle. They can be parallelly arranged. An example is the biceps brachii. There's an asterisk next to that. That might be a test question. They can converge, so these muscle fibers can converge at one point, or they can be pennate, which just means for the strands or pieces of them that come together. So a circular type of muscle would be the Orbicularis oris, which is a circular muscle that surrounds your mouth. A parallel muscle would be an example of the rectus abdominis, or your biceps brachii, which would be parallel muscle fascicle strands. Convergent, you have your pectoralis major there, where it converges at a point, kind of like a triangle. And then pennate have several different bodies to them. Does exercise change skeletal muscle fiber size? Muscle hypertrophy has to do with an increase in fast muscle fiber size, and muscle atrophy is a wasting of muscle that reduces fiber size. We all know that working out at the gym builds muscle, and that means you're increasing the size of the fibers, but not the number of the fibers. Muscle atrophy, if you don't work out at the gym, the reduced stimulation results in reduced muscle size, tone, and power. The words agonist and antagonist have to do with muscles that have opposite actions to each other. So the agonist will be when the contraction of the muscle produces the movement. Also, it's called the prime mover. So an example that they give you is your triceps, which is located on your posterior upper arm. It's the agonist for forearm extension, so it'll be the prime mover of straightening your arm. The antagonist muscle to that will be the muscle whose action opposes that of the agonist. So your antagonist muscle to your triceps will be your biceps brachii because it has an opposite action. Your biceps brachii, when it contracts, will cause forearm flexion, which is bringing your forearm and reducing the angle, as if you were doing a bicep curl. A synergist is the name of a muscle that will assist the agonist in performing its action. An example of a synergist would be a brachialis muscle that helps your biceps flex your arm. Muscle naming. This will be really important when you learn the muscles and where they're located in the body. Muscles can be named for what action they have, where they're located in the body, where they're attached, the orientation of their muscle fibers, their muscle shape and size, and muscle heads or numbers of tendons of origin. Your muscle naming will be easier than bones because you'll see that it just makes a lot more sense, some of the names do, than your bones did. Many of your muscles will have the action in its name, whether it's a flexor or extensor, adductor or abductor. It'll be named for where it's located in the body. So your orbicularis oris will be located around your mouth. Your biceps brachii will be in your brachial arm region. Flexor carpi ulnaris will be around your wrist by your ulna bone. Opponens pollicis has to do with your big thumb. And then you'll have more flexor, extensor, digitorum, superficial or profundus, having to do with superficial or deep. They'll be named for where they're attached. So your sternocleidomastoid muscle is attached to your sternum and clavicle. Intercostal muscles are between the ribs. Fibularis longus muscle is attached to your fibula. Your orientation of muscle fibers, they'll be oblique, rectus is straight, orbicularis is circular, and then muscle shape and size, you'll see deltoid there, means triangular. That's the muscle on top of your shoulder. Trapezius can look kind of like a trapezoid shape. That'll be a diamond-shaped muscle in your back. Gluteus maximus, medius minimus. major, minor. You get the idea that naming the muscles will tell you a lot about where they're located and what action they're doing. We'll finish this PowerPoint talking about the other two types of muscles really briefly, cardiac muscle and smooth muscle. Cardiac muscle, as you know, heart wall. It'll be striated. It forms Y-shaped branches. It's joined by adjacent cardiac muscle cells at what we call intercalated discs. It's autorhythmic, which means it can generate its own electrical impulse without nerve stimulation, which is helpful for maintaining its own heartbeat. And it's also under involuntary control, meaning your autonomic nervous system will influence the rate and strength of contraction of it. So this happens below your conscious level. This is a look at cardiac muscle, and you can see here the cardiac muscle is striated, it's branched, it has the intercalated discs. And then smooth muscle is fusiform and shaped. Smooth muscle generates slow, efficient, fatigue-resistant contractions. And you can see how smooth muscle is not striated. There's one central nucleus. You can see the fusiform shape here. This is a great chart that tells you the general comparisons of all the three types of muscle tissue. And this as well. So your upcoming tests then will be on chapters 6 to 10 on Thursday, March 26th. See your study guide for questions, study your notes. I'll make it available to take on Canvas at 7 p.m. You'll have one attempt, 2 hours to take the test, and I'll leave it open for 24 hours, which means you can take it anytime between Thursday at 7 and Friday at 7. It was great to see you guys, or hear you, or I guess for you to hear me. I hope you're all doing well. Take care.